Thank you very much for joining us this afternoon, this evening, this morning, wherever you are in the world. As Eli was saying, this is something we've run for a number of years now with the WPC, and it's a way of bringing the whole community together around this common condition since 2006 when it came into existence. So it's great to have two great researchers with us. So normally we have one, so two, it's going to be a little more challenging, trying to make sure we get the balance right. So we have David Standard and Karen Yandereis. I hope I said that correctly.
Yep.
Probably not, but it was a good approximation, I would say. Both from the University of Alabama in Birmingham. And we're going to be talking about a topic which is something I get asked a lot about in clinic. I see a lot of people in clinic with this, which is L-DOPA or levodopa-induced dyskinesia. So we're going to have a sort of 50-odd minute discussion about that. But before we do that, I thought it was useful just to hear a little bit about the background of our two researchers today, how they got into research, what led them to work in research in Parkinson's, and how they've got to where they are today. So David, perhaps as I could start with you as the more senior of the two.
Sure. Although I'm not ready to be labeled as senior just yet, I don't think.
I said more senior.
More senior. I'll take that. All right. Well, anyway, thank you for having me today. This is really fun. The WPC is a great organization, and I've been pleased to be a part of it for quite a number of years. So yes, I got in research fairly early on. I was born in New York City and grew up on the East Coast of the United States. And when I was about 13 years old, I got interested in laboratory science. My father was at Georgetown University. He was chair of the pharmacology department. And so starting in about age of 13, all the way through high school, all my summer jobs were laboratory technician. I would ride my bicycle downtown and go work in the lab after school and spend the summers working on lab experiments and really develop sort of a passion for laboratory science. I was an undergraduate at Harvard and got very interested in the brain and neuroscience. And my original idea was actually to be a graduate student and then a PhD investigator. But at that time, I really felt I wanted something broader than that studying just one part of biology wasn't enough for me. And I wound up doing an MD PhD combined degree at WashU in St. Louis. Went from there to University of Pennsylvania, where I trained in neurology and worked with some great movement disorder specialists, Howard Hertig and Matt Stern, who really convinced me that Parkinson's and movement disorders was really exciting and really important, and I should work on this. Went from there back to Boston and joined the lab of Ann Young and Jack Penny, pioneers really in understanding the basal ganglia and how that circuitry works. Built my lab there, was there about 15 years, and then was recruited to UAB in 2006. And the original mission was to build a center, which is now called the Killian Center. It's a group of 19 investigators who work on Parkinson's, Alzheimer's, Huntington's, ALS. I did that for about 5 years, and then 5 years in, the job of chair of the department opened up. And so I've held that since 2010. So what do I do today? I run a large department of neurology. I do see patients every week. I do clinical and basic research in Parkinson's disease. I still feel very much connected to the Parkinson's community. I meet with patients every week and talk with them about how important it is to make progress in this disease.
I mean, just to pick up on a couple of points. I mean, obviously, you were destined for the lab since you went there from a teenager. But what persuaded you that medicine was something you wanted to combine with research? Was it this sense of trying to do something to help people, or was it more about broadening your research horizons?
It honestly, at the beginning, was being broader, about understanding more about how biology works, how humans work. Because graduate school is great, but it does make you an expert on a small area of biology, and Karen may talk about this. Whereas medical school teaches you about everything, right? You need to know about the kidney and the heart and the lung and the liver, and even if you're going to study the brain. And so that's what got me into it. Now, once I got there and started really working with patients and other physicians, I realized, first of all, it was really interesting. It was really important. I like working with patients. And it's a fantastic place to explore new things. there's so much unknown in medicine, especially in neurology, that
Yeah,
it's just every day there's something new in neurology, and I think it's been that excitement that's kept me going. So I'm glad I went that way. I didn't really
yeah,
plan it that way. I didn't set out to be. Honestly, it wasn't a humanitarian goal at the start. It was more of a scientific. This is what I want to
yeah,
study. I want to learn more. But in the end, it turned out to really lead me to the idea that there's so much important work
yeah.
to do in these fields.
And moving into Parkinson's disease, I mean, obviously, you were exposed to great mentors, movement disorder specialists, clinician scientists like Anne Young and Jack Penny. But Parkinson's, why was it you were called particularly to that? Do you think was there something in that
Well,
condition that
similar.
particularly appealed?
Actually, the science got me there first in that.
Right.
So my graduate work was with Dr. Cliff Saper, who you may know he was chair of neurology at Beth Israel in Boston for many years, and I worked on the hypothalamus control of blood pressure and heart rate. And I found I really that way of thinking about the brain as circuits and chemicals that are connected together and talking with each other and this is how the brain works was really fascinating. So when I was thinking about, well, where do I go as a clinical neurologist? Parkinson's disease and movement disorders really is about understanding the circuits in the brain, the chemistry of the brain, and actually changing that. We can change both of those things now, right? We have drugs that
Mhm.
change the chemistry of the brain. We have techniques like DBS that change the circuitry of the brain. So this was kind of a growth of the way I like to think about the brain and study the brain. So again, it began with the science, and then once I got into it, I realized how fascinating this was and how interesting it was to work with people with Parkinson's disease and try and understand their issues and solve the problem for them if I can.
Uh, And you obviously have a passion for it because you still do clinics every week. And so
I
your
do.
basic job at the moment is you have to run an institute, which is a lot of HR and admin, I would imagine.
Yeah,
You,
there's a fair amount of that, raising money, HR, admins, solving problems.
but you still
But
run a lab, David. You, I mean,
I
you're still
do. I still
actively
run a
involved.
lab, and my research has actually migrated into more clinical research
Yeah,
in recent years. We became very interested in the immune system and the role
yeah.
of the immune system in Parkinson's, which is something I know you're interested in. And we could talk for a long time about this.
And we had
But
Ashley on a course this year talking about this.
right. What that led me from was working with Ashley and other people in animal models to more recently really focusing on humans and asking in patients with Parkinson's, what was the evidence for immune activation and how can we understand this? So I do less and less animal model work over the years, and I've done more and more patient-oriented clinical research.
Okay, so that I mean that's great. I mean, I greatly admire people who loyally stuck to what they want to do in terms of really doing clinical work and lab work and informing how one informs the other. So, Karen, can I ask you now about how you got into research, and why in
Sure,
particular have you ended up working in Parkinson's?
sure. My story is very different from David's. I grew up very rural in upstate New York. My dad was a truck driver. My mom was a social worker, and she worked with children. Nobody in my family had a degree higher than a master's degree. Nobody was very sciencey.
Yeah,
So I really don't know how I got here, but I did my undergraduate in psychobiology at Binghamton University, which is a
which university is that?
Binghamton University. It's a
Okay, yeah,
small state university in upstate New York.
yeah,
Um, and it was very heavy on the psychology, light on the biology. And I decided that I wanted to do something with brain imaging. And I had to do a doctoral degree for some reason. I just decided that that's what I wanted to do.
that was in your DNA. You just had to do a doctoral
Yeah, I
degree.
just had to
Yeah,
do it. I don't know.
yeah, yeah.
And then I found out that you were very more likely to get into graduate school if you had worked in a lab. So I asked my academic advisor who I could work with. She said there was a new guy had just gotten there, and his name was Dr. Christopher Bishop. He was looking for undergrads. So I walked down the hall, said, "Hey, I'm looking for lab work experience." He took me in. I was his first student as an undergraduate, and he was
Okay.
doing looking at the role of the serotonergic system in levodopa-induced dyskinesia. And I just started in the lab. I was sat in there all
So
day
when
long.
was this, Karen? So when was this? Did you start working in the lab?
2006, 2005.
2006.
I don't remember
Okay.
really.
So, yeah.
It was the early 2000s.
But
Yeah.
that was very early on. You were involved with
Yeah. hmm
lab work and dyskinesias.
Yeah, I was actually a senior in undergraduate when I started
Okay.
doing it. So
Okay.
yeah, it's kind of advanced in undergrad. But he got me right in the lab, got me working with the animal models. We did a study on ecstasy and
Uh-huh.
it's how it could reduce dyskinesia. And then we did one on buspirone, which is a serotonergic drug.
I think
And I
ecstasy.
actually
There was a big
published
video.
that.
There was a big thing on a video on that, wasn't there? There was a patient, I think,
Yeah, yeah,
took
yep.
ecstasy. I mean, I had many
Yeah,
questions in fact
it
about
was
that.
pretty
Bit
exciting.
not something I could prescribe, but anyway, there we go.
No.
Yeah. So ecstasy and then bus brain. Did you say?
Buspirone, yep. It was
Brain,
a
yeah.
serotonergic 1A agonist. Yep.
Yeah. And so from then, you've just carried on working in this field.
Yeah, so I decided I fell in love with figuring out the circuitry behind levodopa-induced dyskinesia. I fell in love with striatal circuitry because it was so complex and like a fun puzzle to put together. There's lots of moving parts, lots of different cells.
Very good
Um,
moving parts. Yeah.
yeah, not moving parts, but in a figurative sense.
Yeah.
And I just love figuring things out and being hands-on, and it just kind
Yeah.
of fell into my strengths. So
So,
I
and where
stayed
did you
grad
and
school.
where have you done your research over the years? Have you?
I stayed at Binghamton University with Chris as a graduate student.
Yeah.
And then I found out about David as I was looking for postdocs through a fellow graduate student. And I saw him give a talk one time, and I thought it was really cool. And just emailed him out of the blue and told him what I could do and what I was interested in. And he said, "Hey, I have a perfect spot for you in my lab right now." So
Okay.
that's
Terrific.
yeah. And I stayed here since then. So.
Yeah. So that's terrific. Two very different stories. David in the pharmacology lab goes off training in
Yep.
neurology, inspired by movement disorders, inspired by the patients, sets up this whole center in Alabama. And then you have a passion to do a PhD, not quite sure what, but differentiating dyskinesias. And you've been very loyal to that subject for nearly
Yeah.
20. Yeah.
Yeah.
So let's pick up on L-DOPA-induced dyskinesias, which are obviously a very common problem. There may be people on this call with Parkinson's who are very familiar with this. So perhaps I could ask you, David, I mean, perhaps you could just give a brief sort of description on what people mean by L-DOPA-induced dyskinesias, how common they are, and are they a problem?
Well, yeah, I think this is one of the problems we really wrestle with in the clinic. You know, L-DOPA, I always say, is the miracle of Parkinson's disease. It's amazing that we have this pill that you can take that gets into your brain and replaces the dopamine that's missing and can actually make people better. I mean, this is extraordinary. We
Yeah.
don't have very many other diseases where you can just take a pill and it fixes the problem in the brain. So levodopa is amazing stuff, and you know, the history of this is. You know, this has really been with us as a clinical drug since the late 1960s, and is a very powerful treatment and really helpful to a lot of people. But the limitation that was realized early on, even in the 1970s, they realized that if you took enough levodopa for long enough, you very often develop these abnormal movements, which we call dyskinesias. So these are the writhing movements, sometimes starts as just kind of a little bit of rocking, but then can become much more violent and much more dramatic. Many of you have probably seen Michael J. Fox, some of his interviews, and actually his movie is a really nice movie called Still, where he demonstrates his dyskinesias. You can see them very dramatically in that movie. And, you know, they can really become disabling. The dyskinesias can become the big driver, right? So when they start and they're mild and people are just exhibiting a little bit of movement, I tell people, well, let's not worry about it. You know, that's not enough to get excited about. But when it starts to be enough that you can't walk, you can't eat, you're falling down, and the unpredictable nature of the dyskinesias, I think it's really troubling to people that, you know, they usually come when the drug is highest in your system, but not always. And sometimes that's hard to say when that would be. I've had people pulled over by the police on the highway because they had dyskinesia driving behind the wheel, and the police were
Yeah.
concerned.
Yeah.
Actually, at one patient, I wrote him a letter to take with him because this happened to him several times. And I said, in the letter, I said, just let him sit and calm down for a little while and this will go away.
Yeah.
And he used that and showed it to the police on several occasions.
Yeah.
But, this can really get to be quite a severe
Yeah.
disability. And it is one of the major reasons we move on to treatments like deep brain stimulation, which is
Yeah.
an effective treatment, but very invasive, obviously.
Yeah.
How many people get dyskinesia? If you look at populations, at about 5 years, 50% of the people treated with levodopa will have some degree of dyskinesia. And at 10 years, you're probably talking 80 to 90% have some degree of dyskinesia. And in many of them, it's quite disabling.
Yeah.
And our
And I, and
ways
I,
of,
and I often say it's a sort of it's disease duration plus dose of medication. I don't know whether that's a true, if you give people industrial doses of L-Dopa in the first year, they'll be dyskinetic. I think in the L-Dopa study
I
they
think
showed.
that's generally true. There's a lot of debate about this,
Yeah.
but the duration of the disease does matter.
Yeah.
And I think that probably actually what really matters is how much of your own dopamine system have you lost?
Yeah.
Because we know in an animal model that's really critical in producing dyskinesia, and Karen can talk about that. So,
Yeah.
how severe is the change in the brain? And then how much levodopa are you taking? And also how are you taking it? Are you taking it in, very large doses, a few times a day? Or are you spreading it out? These things do seem to make a difference.
Yeah.
So I've been working with solutions for this for a while in the laboratory, trying to understand, well, why do you get this dyskinesia
Yeah.
anyway?
Yeah.
What's going wrong in the brain that you're inducing this? And I was struck early on. Actually, I came to UAB and was talking with some of the memory researchers here, and really started to think about dyskinesia as a kind of memory. In other words, you get dyskinesia because your brain remembers that you've had this levodopa before.
Yeah.
And that's in some ways very similar to drug addiction, right? Well, when you take cocaine, your brain is responding because it knows it's had this before.
Yeah.
And you get addicted because the brain gets, really becomes dependent on that. I don't think levodopa is addictive exactly, but this motor response doesn't come with just one dose of levodopa. It comes from that repeated
Yeah.
administration, the brain building a memory that it's seen this before and now it's going to overreact to it. So
And
that's really,
and if you give it continuously, even in people who've got this, I mean, you can smooth them out. So
yes, if you give
would
it
that be a fair
very
statement?
continuously, and this was done experimentally in the 90s, Tom Chase and Maral Moradian at the NIH actually used intravenous levodopa, which is very difficult to use.
Yeah.
It's hard to get, very difficult to use, but they did it at the NIH and were able to show if you gave intravenous levodopa and really leveled the levodopa
Yeah.
concentration out, it improved. And now the technologies we have are things like Duopa, which I've worked with a lot, which is intestinal levodopa, and
Yeah.
more recently subcutaneous levodopa like Vyalev. And these do reduce dyskinesia as well. And
Yeah.
so they
Yeah.
they're not perfect at it, but they
Yeah.
do. The more continuous delivery seems to reduce the severity of
Yeah.
dyskinesia.
So I mean, I guess just to sort of summarize, so L-Dopa-induced dyskinesias are almost inevitable in everybody with Parkinson's taking L-Dopa, and I think it's important to distinguish between the sort of mild ones where people have a little bit of head movement and a little bit of often on the side where it began, versus those where it really does become a major
Right.
disabling feature of their condition.
Yeah,
And
I often tell people, if there's you get two different stories, right? Sometimes if you talk to the person who has Parkinson's, you get one story. If you talk to their
yeah,
family, you get a different story. The family is often concerned even
yeah,
about these mild dyskinesias. And my
yeah,
usual answer to that is most patients with Parkinson's I've spoken to actually feel good with a little bit of dyskinesia. This is loose, I can move, I feel
yeah,
well. And what I don't want to have is I don't want to be stuck. I don't want to be frozen. I don't want to be immobile. That feels terrible.
yeah, yeah,
Whereas the family thinks they look better when they're not moving. So you have to do some education here to explain that the person with
yeah,
the dyskinesia actually may feel fine with a little bit of that, and I don't worry
yeah,
about that. But it's
yeah,
when it becomes violent and
yeah.
disruptive that it's really a problem. And that happens in a significant number of people.
Yeah, and I certainly remember early on in my career having a sort of domestic in the clinic where the patients often aren't necessarily aware that they've got the dyskinesias, but
Right,
the wife was absolutely adamant that she hated this because it made her feel very uncomfortable. He was absolutely delighted not to be off, and the two of them
right,
had a very
right.
different perspective of what they wanted. So you're absolutely right. I think trying to understand why they occur and explain to people, who's actually got them and what they mean for them. So I suppose we could say they're inevitable with Parkinson's. They go through this gradation from being there but not particularly troublesome to becoming very disabling. And this unpredictability, which is often what is a very disabling aspect of the condition. You can't plan your life because you don't know what's going to happen. So this is something which happens in the clinic. So perhaps I can come to you, Karen, and say, but my understanding is the only animal on the planet that gets Parkinson's is us. And so L-DOPA-induced dyskinesias are by definition only going to be something you see in us, or people with Parkinson's. So how can you model this in the lab? I mean, what's going to be the model for dyskinesias in the lab? And when you started all those years ago, what was the model you used, and how have they changed and been informed by what we see in the clinic?
Sure. So the simple answer is it hasn't changed much since the 70s.
Okay.
Really, the model we still use to study levodopa-induced dyskinesia is the classic 6-hydroxydopamine model, which is a neurotoxin that's kind of selectively kills off dopaminergic cells in the brain. And the way we approach this in the dyskinesia field is we infuse 6-hydroxydopamine into a bundle of neural fibers that runs from the area that has the dopamine neurons, called the substantia nigra, to the striatum, which is part of the basal ganglia that is kind of the movement control center.
Yeah. So you do this in rats typically, do you?
Rats and mice. We typically use mice now because we're doing genetic studies. Yeah. But originally I did this in rats, and that is kind of the preferred model for behavior. But you do this on one side of the brain so that you can have a control side within the same animal. Yeah. And you allow the dopaminergic lesion to process for a few weeks. And this leads to a condition in the striatum where it develops like supersensitization to dopamine. And then when you give a dopaminergic drug, after the supersensitization progresses, it results in hyperkinetic movements instead of just fixing the Parkinsonism. It leads to hyperkinetic movements, which is
basically you have a rat or a mouse. You have one dopamine system that's intact. You completely kill the dopamine system. So you're mimicking the dopamine loss of Parkinson's. It's not Parkinson's, but you're mimicking the dopamine loss. Right. And because the receptors on this side, which have no dopamine input, are saying, "Where is my dopamine?"
Yes.
When any comes along, they think that's terrific, but they slightly overreact, and then you get overstimulation, and that produces involuntary movements. Is that the sort of idea of the model?
Yep.
And how do you stimulate those dopamine receptors? How do you make that system overreact? Do you give them the drugs you use in the clinic?
Yeah, same drug, levodopa. Mm-hmm. Yeah. Or you can also use dopaminergic agonists, like I can't think of any of the specific ones. David would know the name.
One I always use.
Pramipexole or ropinirole are the things that we use in the clinic, and in an animal model, there are other drugs that are easier to use in a rodent, but they work the same way.
Mm-hmm. Okay, so basically, you give these animals L-DOPA like you would an individual with Parkinson's, and in this unilateral one-side lesion, after a while, those drugs, whilst they make the animal better, I presume, they get rid of the asymmetry.
They do get rid of the Parkinsonianism. They do get rid of the Parkinson disease symptoms, but they then slowly progress into dyskinetic movements instead. So it's kind of like.
And are they easy to see? Are they easy to see in the animal? I mean, do they have these sort of writhing movements?
Yes, and it looks very similar to what you would see in a human. Yeah.
And are they on the other side from the lesion, or do you see them on both sides?
Just one side. Yep.
Just one side. So
it's very easy to see. Yeah.
Okay. And so when you see that in your animal model, so how do you take it from there? So do you then, I mean, one of the problems in the clinic, as David alluded to, is you can use these advanced therapies, but obviously you'd like to do something before that. And the only drug, I don't know what it's like in the States, but in the UK, the only drug we really have is amantadine. Right. That we can use to deal with dyskinesias. So if you give your animals amantadine, do the dyskinesias go away? So you can sort of say, actually, this is quite a good model, because I give the drugs, they get dyskinesia. I give the drugs I use in the clinic, they go away. So this is a model which we can rely on for clinical information.
Yeah, so a lot of the same drugs that reduce dyskinesia in clinic also reduce it in this animal model.
Okay.
So drugs like amantadine, the serotonergic drugs that are now being developed to use in the clinic also
Okay.
work in these models, and were developed using these models. So
I mean, obviously you have this abnormality. So how do you then dissect what's causing the problem? So the simple argument used to be that the reason you get dyskinesias is you give these drugs, they come out in a great dollop because you swallow them, they get taken up. You get this big dollop of dopamine, then it disappears. And in the early stages, that doesn't matter because you can buffer it. But after a while, that dollop of dopamine overstimulates the system, and it's that pulsatile release that's causing the problem. But I don't know whether that's a view that people would subscribe to. And then if it is, what is it that goes wrong? Why is that pulsatile release such a bad thing? And
yeah,
I want to take it on to David's thing about engrams, because that's quite interesting. I haven't.
Well,
Yeah,
Karen
so
gets credit for the engram. I never pushed it quite that far.
no, he
To
was
talk
very
about
conservative.
engrams. But you're getting at the question here, which is when the first time you give a mouse levodopa, you don't get dyskinesia either,
Yeah.
right? And the first time you give a human with Parkinson's levodopa, you don't usually get dyskinesia. But if you give, in a human, it takes several years to see the dyskinesia usually. In a mouse, we can see it in 5 or 6 days, but
Okay.
it still takes repeated dosing. So what is it about that?
Now those big
What's
doses,
going
David.
on there?
So
Hmm?
in
No,
comparison to the person, are they big doses or they're just a sort of normal? So they're not big doses. So low doses in a completely dopamine annihilated system, you get dyskinesias within the week.
Yeah, and this goes back to the point you made earlier, which is it's something about the degree of dopamine loss is very important in this, right? So if Karen's great at these mouse models, she's really good at it, and so her mice always have almost complete loss of the dopamine system. But
Yeah.
if you don't do it quite right and you only kill 70% of the dopamine neurons, then the mouse won't get dyskinesia. Just as
Yeah.
in a human with early Parkinson's, you don't usually see dyskinesia. You see it after the dopamine system has really lost a
Okay.
lot of its activity.
So that's the real key thing, the degree of loss of the dopamine system,
Mhm.
obviously with the drugs, because you wouldn't get it if you didn't give the drugs. So Karen, so when you've got this model, I mean, how do you dissect? What's the next thing you do in terms of trying to understand what might be driving
yeah.
dyskinesia?
So what I think is really cool, well, not cool, but interesting about dyskinesia is that it's really progressive. So it gets worse over time. And part of that
Yeah.
is the disease, but also even in mice, when we give the same dose over days, they develop dyskinesia with the same dose. So it's not just the cell loss.
Okay.
And
So if
then
I gave it to
other,
them in week 1, they would get a degree of dyskinesia. If I gave them the same dose the following week, they would have
right.
even more dyskinesias.
Yes.
So
Yes.
it sort of gets ramped up every time.
Right.
So it has more of an effect every time you get. Okay. Yeah.
Right. And then the other interesting aspect is its persistence. So in an animal model, if you take off levodopa after they've become dyskinetic, if you take it off for a week, a month, and you give them levodopa again, they have the same degree of dyskinesia as they did before. So there's kind of this, what David was saying is there's kind of this memory for dyskinesia that is somewhere, we don't know where, in the brain.
Okay.
So kind of riffing off that memory idea, if you go into like learning and memory literature or into drug addiction literature, this new idea is the engram idea. So it's
Okay.
basically this idea that there's a cellular memory for prior experiences, and that certain groups of cells kind of hold this memory within them, and they're called engram cells. And
Right.
whenever they experience a similar stimulus or a similar experience, again, that facilitates the same type of response over and over, and it facilitates the response being bigger, and it facilitates the response being faster. Okay. So it's
So you
kind of,
learn something across a synapse. So would this be similar to things like post-traumatic stress disorder, where you've had a traumatic experience, you've got a memory for it, and then
sure.
when you encounter something similar, you get a magnified? Is it that sort of principle that somewhere in your brain
Mhm.
you've wired together an association
Right.
that's there and it's laid down, and you're not going
Right.
to get? Yeah.
Yeah.
Okay.
So our kind of question when David presented this idea of a memory for dyskinesia, I was really interested in that, and then started reading that literature. And it turned out that there was a lot of evidence for these so-called engram cells.
Yeah.
So the question to us was, okay, what are they? What are these engram cells? There was a lot of evidence from prior research suggesting that they were one of the main output neurons in the striatum called
Yeah.
medium spiny neurons, and not just any medium spiny neuron. It was a specific subset of those called D1.
So this is what the dopamine fibers from the nigra
Yeah.
synapse onto these medium spiny neurons.
Right.
They
Yeah.
would be in some ways the obvious candidate in the sense they've lost that input, and so is
Right.
that where the number is being laid down. So can I ask you what's the normal point of it? So
What's
they
the what?
they become abnormal for dyskinesias, but are they there in the normal brain, if you like, and are they doing something to do with our normal motor behavior? So do we learn? Do we learn
Yes.
in our striatum all the time?
Oh
And
yeah.
that has a very
so-
beneficial purpose. So it makes Tiger Woods play and win golf very well. Or
Yeah.
is it, but when it's in a pathological state, it drives abnormal behaviors. So what does it do in a normal striatum? And then we'll come on and talk about it in dyskinesias. I mean, we might
Sure.
not know, but I'm interested.
Sure. So the striatum is really a very interesting brain region to study because it has kind of two subsets of neurons. One is called the direct pathway neurons, and the other one is called the indirect pathway, and they're both medium spiny neurons, but they do vastly different things. They have different outputs and inputs, and they do lots of different things. But in short, the direct pathway, or the D1 MSNs, they're kind of like the gas in a car. They
Right.
kind of push you towards acceleration of movement or towards movement. And the other half of it, the indirect pathway, or D2 MSNs, they kind of are the brakes. So they have
Okay.
kind of a suppressive effect on movement. That's a very simplified way of talking about it.
So the
But
D1 are pushing you, the D2 are putting the brakes.
yeah,
So both of them working together to get the right acceleration to acceleration and get the
yeah.
right.
So you have this balance in normal movement
Yeah.
acquisition and normal movement selection
Yeah.
to have this balance between both pathways. So when you want to learn something new, the indirect pathway is kind of silencing those behaviors that are competing with the behavior that you want. Does that make sense?
Okay. Yeah, and I guess the dopamine is what's regulating that. So once that's
Right.
got,
Yeah.
these populations start to operate a bit independent of each other.
Right.
Yeah, I mean, the other way to think about it is, and this is the analogy I often use, you mentioned Tiger Woods, right?
Yeah.
So he plays golf well. I don't play any golf.
Yeah.
But if I was to decide to take golf lessons, which I don't really have time for, I would go out and look at the ball, and someone would hand me a club. I wouldn't know how to hold it. I wouldn't know how to swing it, how to stand, all these things. So each one of those individual actions, I would have to have a coach to talk me through it and think through it. And how do I stand there? How do I hold this? How do I swing this? Where does the ball go? But if I did it 1,000 or 10,000 or 100,000 times as Tiger Woods has done, you don't think about that anymore. You just go walk up to the ball and hit it. And it's a program. You've gone from a bunch of individual steps that your brain was doing to, I just have a program. I have a program that I run when I hit the ball, and I don't
Yeah.
think about where I'm putting my hands or how I'm standing. It's all just baked in. And that's
Okay.
what the basal ganglia are doing. They're taking those individual actions and baking them into programs. And dopamine is part of that learning. So this whole idea of learning and dopamine is very much a part of the normal function of the basal ganglia. And then when you start throwing big doses of levodopa into the system, in a system that lacks dopamine, you get abnormal learning. And that's really how I
Okay.
think of dyskinesia, as a kind of abnormal movement that your brain has learned under the influence of all this levodopa.
And one of the questions which has come up is we talk about the dopamine system as obviously the major issue. But one of the questions which has often occurred in the area where I work around transplantation and dopamine cells is the serotonin system, which is obviously a different group of neurons originating close to the dopamine cells in the brainstem. They project into the striatum. And so, the extent to which these non-dopamine cells, which are affected in the disease process, but more intact, are they contributing? So do they take up L-DOPA, release L-DOPA? So you've not only got a failing dopamine system, you've got pulsatile dopamine
Well,
coming out of
you
it.
just asked
You've also
Karen
got
for the topic of her PhD thesis.
Yeah.
She did much of the work on this.
Okay.
Yeah, so along with Angela Chinchi,
Yeah.
I was one of the labs that kind of pioneered all that research in the serotonergic system and its role in dyskinesia. So yes, when dopamine cells are lost, serotonergic neurons can step in and take over for the role of dopamine neurons in releasing dopamine into the striatum. So,
But they can't take it back up. That's a bit of a problem.
no, yeah. So that's part of the reason why we think that dopaminergic release induced by L-DOPA therapy is kind of dysregulated, because a lot of it is released by these non-dopaminergic cells that don't have systems to retake that dopamine up, and they don't have a brake system. So they don't have a auto-regulatory system to tell them that, well, wait, that's too much dopamine. They don't have that. So they just
So basically,
out.
so just so I've got it clear in my head. So as you start to evolve through the condition, the dopamine system fails. Your brain can cope with it. Your striatum, your medium spine neurons, all right. But after a while, you think, actually, I'm starting to lose the control here of my hand, my foot on the accelerator, on the brake. They're becoming a little erratic. And superimposed on that, it's not only have I got a failing system with dopamine, but I've got a serotonin system that's pumping out dopamine in an unregulated fashion. So I've got I've got this very dysregulated system with neurons which are dependent on quite a fine tune of dopamine to maintain a normal control. And now that's starting to go wrong. And so I'm starting to develop the dyskinesias. And if I've understood it correctly, you're then saying that going wrong now tells your medium spiny neurons, this is the new memory. So you learn to do that. But now I need to learn to do that. So every time you see this is what's going to happen. And so you've now put your memory down for something that's abnormal.
Yeah.
So every time you see that drug, you're going to do the behavior which you've learned, or that bit of the brain, and that's the dyskinesia.
Yeah. So the real question was, what is kind of holding that memory for levodopa? Like, what cells are they, and how do they hold the memory? And that's kind of our driving force behind our work for the last, 5 years now. And the way we kind of approached it was kind of to figure out which cell in the striatum is kind of driving this. We used kind of a new technique called single cell RNA sequencing. And it's basically a way to tell what every cell, or a certain percentage of the cells in a certain brain area, what proteins they're actively making. And from looking at which proteins are being actively made, we can infer what they're doing. So we use that technique kind of as, it's kind of a fishing expedition, some people would call it, but we looked at which cells in the brain were reacting to L-DOPA.
Right.
What were they doing? So that was kind of the first step.
And what did you find?
So
were you
we
right,
found
it was the D1, it was your D1 medium spiny neurons where the problems lie.
Yeah, so it was those direct pathway D1 medium spiny neurons, but it wasn't all of them. So it's not like every single direct pathway neuron was doing the same thing. It was a subset of them that were reacting to L-DOPA differently than all the other ones.
Okay.
So
why is that?
Well, that's a great question. And that's what we're still trying to figure out. But in general, we think that it is a process called epigenetics. Epigenetics is kind of this interaction between the environment and genetics, and it's the way that your body decides which genes to turn on and which ones to turn off. And what we noticed when we looked at these what we call these engram-like cells, these D1 MSNs, medium spiny neurons, when we looked at them, we saw that they were expressing a lot of proteins that were involved in epigenetic processes, especially ones that were involved in opening up the chromatin. So chromatin is like this three-dimensional wrapping of your DNA around it's kind of hard to explain in layman's terms.
Well,
But
it sort of compacts it, doesn't it? It keeps it all very
yeah,
compact, it keeps it
yeah,
tight, so the DNA can't misbehave. You can't transcribe
right.
off it. It keeps it silent in some ways.
Yeah, so in an adult, most of the genes are turned off. So
Yeah,
in an adult neuron, most of the genes are turned off. If they're opened up, and there's a lot of them opened up, it's
yeah,
kind of what you see in cancer. Okay,
yeah.
so you want to keep your DNA really tightly wound, and you only want to unwind it when you want to make a certain protein. So what
Okay.
we noticed in these like engram-like cells is that they were unwinding certain areas of their chromatin or their DNA to make them newly available to be made into proteins.
So
And
basically,
so, yeah.
so carry on. Sorry. Yeah.
So we're using all of these techniques like a single cell RNA sequencing to learn this, and then we used a new technique called single cell ATAC. I'm not going to tell you what it stands for, but it's just it just lets you it gives you like a whole DNA kind of view of which areas are open. And then we look at what factors are involved in opening up that chromatin.
Yeah.
And from this, we think that we can kind of turn back time a little bit by erasing those proteins that come in to open up the chromatin. And
Yeah.
that if we can erase those open areas, we can revert back to that first time you saw levodopa when you were early on in Parkinson's disease. So that's kind of the main view. We're still kind of in the middle of that process. But that's
So the
kind of
hypothesis,
cool.
so the hypothesis, just so I get it clear in my head, because this is quite conceptually.
Yeah.
Essentially, what happens is you start to learn something that's abnormal, and the way you learn is that the cell says, "Well, I want to change my response, so I want to code for new things in order to now cement that." So with a memory, so if you're keeping a memory, you say, "That's a really good memory. I want to open up my DNA. I want to produce some proteins. I want to glue that memory into my cell, and then I've got it forever." And here, of course, it's doing it in an abnormal situation. So when the chromatin is opened up, what sort of new, so the RNA coding for the proteins, what sort of new proteins are being made in the cell, which makes it remember? Because presumably it's then amplifying some sort of response. So what are the type of proteins? Do you know what the proteins are that are being made in these MSNs, which are enabling them to remember this abnormal behavior, if you like?
Yeah, so it actually varies depending on when you're looking. So early on in dyskinesia, as it's developing, like before you even really get really bad behavioral response, before you get the really bad dyskinesia, you get upregulation of these epigenetic proteins. They're called AP1 factors. And these AP1 factors are really the ones that are opening up other areas of the chromatin.
Okay.
And then later on, as dyskinesia develops and it becomes more progressive and more persistent, you're getting proteins that are more involved in remodeling the whole neuron itself. So they're creating new connections, they're solidifying the connections they have formed. So it's kind of like making the computer program for dyskinesia stronger, if you want to kind of view it that way. So they're making new proteins to allow for new synapses, for strengthening of those synapses, and for kind of pushing the cell to change in its conformation and how it communicates with other cells.
And so, so this is, so basically a sort of rewiring how these cells communicate with other cells.
Yeah. Mm-hmm.
Early stage is basically opening up. So one argument, if you could catch it in that early stage, when you've merely opened up the DNA, but you haven't got it, it hasn't actually cemented its new connections. If you could shut the chromatin down at that point,
right,
you could stop the dyskinesias from ever developing.
Presumably, yes.
Yeah. And so one of the questions I would have is that sounds very nice to do, but from my limited understanding, going after chromatin is not a simple thing to do in terms of shutting it down and opening it up. And drugs like amantadine, which we mentioned earlier, they're not working in this way. Amantadine presumably works by modulating dopamine release. You're going to have to find a new agent.
Yeah.
Work on this sort of epigenetics. And the only drug which I ever know about this is sodium valproate, because that's always been shown to have something in that area. But I'd be interested to know, how do you now, having sort of found the mechanism or a possible mechanism that could be involved, how do you take that forward therapeutically? Because there'll be lots of people on this call. I've got lots of questions here in the chat about, I've got these type of problems. I've got facial dyskinesias. I've got this. Younger patients have more dyskinesias. So the critical question I'll be asking is, that's great, Karen, but what does that mean for me? How can I turn off this epigenetic problem?
Yeah, so there's kind of two different ways that you could probably do that. One of them is to reduce D1 receptor activation. So the way that you would do that is through drugs like amantadine or the serotonergic drugs that reduce that signaling onto D1 MSNs. The
idea then would be presynaptic. So basically stop things coming and stimulating.
Stop the excessive dopaminergic signal.
Yeah. And that's presumably how these continuous treatments help in some ways, because you've sort of leveled that out. Yeah. Okay. So that's one way. Yeah.
So that's one way. And the way that I'm envisioning it in the future, and this is way far in the future. So is kind of using CRISPR-based strategies to edit which areas of the chromatin are open. So in theory, we could target specific areas of the chromatin using CRISPR-based strategies to
so these are gene editing strategies. CRISPR's a way of actually with great skill and precision actually editing bits that you want to edit.
And there's a lot of tools being developed that are way more specific than anything we have ever developed before in terms of that. So that is, of course, far in the future, but that's the way that I envision our work contributing to the field in the future.
And so in order to do that, you would have to presumably develop something that's pretty selective, which you would then inject into the striatum. Would that be, because I can't imagine you could give something orally or intravenously.
Well, I mean, by the time that we get to that point, there may be some new strategies in that way. But yeah, at this point, CRISPR-based strategies are mostly intracerebrally given. So, and I don't even think they're really doing that yet. So.
Well, CRISPR, not yet, but imagine if this worked, right? If you could basically erase the dyskinesia, someone who's had Parkinson's for 7 or 8 years, they have really troublesome dyskinesia, and with say, one treatment, even if it had to be given into the spinal fluid one time, it would erase the memory of dyskinesia and reset the clock. And you'd have another 5 or 7 years before they came back. I mean, this is kind of it sounds like science fiction, but lots of things we do today were science fiction 10 years ago. So, I don't think it's impossible to think about those things.
Yeah, well, I think you're absolutely right. I mean, I think,
sorry.
I was going to say, well, I think, when I started in medicine, the idea that you'd put an electrode in someone's head and stimulate it at high frequency and that would deal with Parkinson's, you think, well, that's completely bonkers. But actually, it's pretty effective. So, but in your models, Karen, how are you approaching this in terms of trying to tackle this epigenetic? You've obviously got this ATAC sequencing, your single cell. So you're describing the problem. So you can describe the chromatin, these epigenetic changes. You can see that there's changes in proteins. How do you then sort of try and prove causality? So how do you say, well, I've got lots of changes. How do I know those are the critical ones? And how does your CRISPR technology work in that? Can you do that in animal models, or is it actually?
So we haven't tried any of the CRISPR yet. We're still trying to figure out which areas of the chromatin are open and which ones are driving the behavior. So that's where we're at right now. So I can't really speak to how we're using it right now, but that is kind of where we're moving in the future.
And when you say it's only a population of these cells, do you have any ideas as to which population it is and what that might mean for people? So is it the area with the greatest dopamine loss? Is there a subset of D1 medium spiny neurons which actually are quite well defined? When you get a human post-mortem tissue, you can sort of say, actually, I can see these cells. And if you look at post-mortem tissue in patients who died with it, can you, I mean, I don't know whether it's possible, people who've got bad dyskinesias, people who've not, can you see similar changes in the human brain using these technologies, or is that not really viable?
I don't know of any human studies. Maybe David might know better, but as far as why or which ones are more likely to become these engram-like cells, we don't really know yet. That's kind of what we're trying to figure out. And it's probably something to do with the dopaminergic cell loss and the dopaminergic input loss to the striatum. So most likely the ones that are kind of recruited into this engram are probably most likely the ones that are most affected by the dopamine cell loss. But in reality, we don't really know, and they seem to be kind of spread throughout the striatum. It doesn't really seem like one specific area is more than another. So we don't really know. We have no idea.
It's a subpopulation which is sort of scattered throughout that for some reason have this rather unique role. So it's not, there's a subpopulation where this is really what's driving.
Right. So in the learning and memory literature, there's a lot of evidence that suggests that the cells that get recruited into this like pattern of behavior are already have a more open chromatin, and that there's like cycles of open and closed chromatin as you go throughout your day and as you go throughout your week, and that those cells are just more ready for a stimulus to come to activate them. So there is evidence that that is out there, but that's very new evidence, and we haven't seen if that's why it happens in dyskinesia.
And in drug addiction, obviously, some of the problems, so with the dorsal striatum is what's affected in Parkinson's and the dopamine loss there. The ventral striatum, from what I remember, has quite a lot to do with drug addiction, and that also has quite a lot to do with dopamine. So is there, I mean, you sort of intimated that there's a literature there on drug addiction with dopaminergic drugs. And is it a similar hypothesis there that there are a population of engrams in the ventral striatum, which now are activated by increased dopamine in certain individuals? And then the question would be, why do you only see it in certain individuals with drug addiction, whereas you see it in everybody with POC disease? Is that because in people with POC disease, they always lose their dopamine system, and there's some subtleties in the dopamine system of drug addiction?
Well, just to be clear, I'm not sure we see it everyone with Parkinson's, right? And you've had this experience that some people take large doses of levodopa for many years and don't get dyskinesia. That's not the average result, but there are individuals like that. And there are people who take actually relatively small doses of levodopa and get dyskinesia quite early. So there is a range of sort of human experience with this. And I think that there's probably underlying genetics here. We know some of the genes, variations in a gene called COMT seem to control dyskinesia risk. There's been a lot of things in the chat about men and women do seem to have more dyskinesia than men. Now, whether that's because the body weight difference or an actual biological difference, still kind of unclear. But there's variation, just as there is in drug addiction. Not everyone is as prone to drug addiction as the next person. Some people seem to have a very high, and you can see this with alcohol too, right? That there are genetic tendencies towards alcoholism. So there is variation in the human population. I think this is true of dyskinesia, that some people are more sensitive to all of this than others. But on average, at 5 years, half of the patients with Parkinson's will have dyskinesia. So it's quite common.
Yeah, no, it's a good point, David, because I do have certain people in clinic who, 10, 15 years into cognition on fairly high doses of medication. Dyskinesia is just never, I mean, I assume they've got Parkinson's, but I mean, they never seem to emerge in some people. And I mean, there is a question about young onset being slightly more predisposed to getting dyskinesias. Do you think that's true? Probably perhaps David's best answer. Do you think it's true that younger onset patients get it, or do you think younger onset patients, because they have longer ahead of them, or they've already had a more dramatic dopamine loss because they presented earlier? I don't know, David, whether you have a...
In younger patients, I mean, they do tend to have a fairly dramatic loss of dopamine in the striatum. And so that may be the reason. And they do tend to respond very well to levodopa. So they often get on it early. Whether that alone is the whole explanation for why they get a lot of dyskinesia in young onset, or whether there's more to it, I don't think we know.
Yeah. Okay. I mean, we're coming towards the end now. So in the last minute or two, I mean, someone's asked a question about funding, which it's an interesting question. But I suppose one of the questions which does arise from this is, and certainly Angela has always bent my ear slightly on, is dyskinesia does it get enough funding and does it get enough research? Because you can always argue that at the end of the day, we can treat dyskinesias because we can give infusion therapies and we can do deep brain stimulation. So I suppose my final question is how important is it for us to really understand the basis of dyskinesia? Not only to really understand dyskinesia, but to understand some fundamental aspect about how basal ganglia function or how motor control is organized in our brains.
Well, I would say, motor control is really interesting, but, treatment of dyskinesia, amantadine is okay, but not that great. Let's face it. I mean, that is not the answer to the problem. And this problem really troubles a lot of people. It also holds us back from using levodopa more earlier in the disease. We would use more levodopa if we had an easy fix for dyskinesia. So I think it's a real problem. In terms of getting this research funded, I mean, Karen's been pretty successful. She's gotten funded by Department of Defense and now by NIH, but that is she's an outlier in that area, I would say. She's been lucky with that, but it's tough and convincing actually the people who support research that this is an important problem is one of the real challenges in this area.
Yeah, I mean, I completely echo that. I mean, I would say that in the clinic, if we had another, so I always say that, amantadine works quite well, but it's the only thing we've got. And if you don't respond to amantadine, you're only looking at these. So I think, if you could get something that could help gait, which was where I was talking to Lynn Rochester last time, and if you get another agent that could help with dyskinesias, that would be a major advance. So, I mean, our time is running out, but I want to thank you both, David and Karen. I mean, I've personally found that incredibly stimulating because it's a sort of very interesting new idea. It's wonderful how you've pursued it and developed your theories on it and how you're tackling it and the potential that offers. I think not just for treating dyskinesias, but actually how we understand how the brain regulates its activity, how it stores information, how it changes activity, and the plasticity of it, which is clearly the case in more aged brains, which for David and I is a very useful thing to know that there is still plasticity in there, which might be helpful. So thank you both very much for a very, very informative chat this afternoon. I would like to thank Supernus Pharmaceuticals for their support in making this series happen. That is it.